Hi, I'm Carpenter, and it's my pleasure and privilege to be here representing the JUMP-Cell Painting Consortium and telling you about what we've been up to. First, I want to make sure we're all on the same page with respect to understanding what cell painting is. It's this assay that we're using that labels a variety of organelles within the cell in order to really just extract as much information as possible from the morphological state of cells. And the goal, once you have that information, is to predict assays or to match these profiles from drugs to genes to disease states. And in fact, this general approach of cell painting and of image-based profiling more generally has been proven to be useful up and down the drug discovery pipeline. So here's some examples shown here along with some references from my own lab's work and from some other labs' work, but really there's tons more out there demonstrating that this is a useful kind of data and a useful way to interrogate cell systems. The JUMP-Cell Painting Consortium, JUMP stands for Joint Undertaking for Morphological Profiling, and this group of partners has gathered together in order to accomplish these goals. First, and probably most dramatically, to make this giant public cell painting data set across all the partners, but we also want to create a community and develop best practices and share them among each other pre-competitively. So for the pharma companies that are involved, the goal here is really to get on board and to adopt the best possible cell painting techniques within their groups so that the whole community can really accelerate here. And we also want to be sure that we can align this public data set that we're producing with any future partner-produced data. So let's go through these one by one. First, the data set itself, which I hope many of you are excited to eventually download and work with. It's going to be 150,000 perturbations in 5 replicates each. And the contributors and partners are shown here along the left. We're going to, each partner is contributing generally 12,000 compounds or genetic perturbations worth of data. And the kind of data that's going to be extracted here includes both classical image-based features from Cell Profiler as well as deep learning features. And we expect to deposit probably several versions of each of these over time, as well as the raw images to become publicly available. So what have we accomplished so far? So far, the partners have selected the compounds that they want to test for the most part, and they've decided on compound exchange logistics. So this is a really unique property of this consortium. It would be easy if each company just picked 12,000 compounds, ran the 5 replicates internally, and then deposited the data. But in reality, each company is going to do 1 replicate internally and then produce 4 aliquots of the compound to send to other partners. And those other partners will be running the other 4 replicates. By having this crisscrossing exchange of compounds and having data that's produced at 5 different sites, we really hope to have sufficient data to figure out how to normalize and align data that has been produced at different sites so that the public resource, as well as each company's internal future data, is as aligned as possible. We also are creating this community, getting to share information about what works best with each other and develop best practices. So we are also carrying out a very extensive series of pilot experiments. And those pilot experiments aim to optimize assay parameters such as the reagent concentrations, microscopy plates, cell types, time points to be used for the assay. And we're also comparing genetic perturbations against each other. And for a very specific purpose, our goal is to see whether what type of genetic perturbation, whether overexpression or knockdown by synthetic or lentiviral CRISPR, which of these is best able to produce signatures that match the compound signatures for cases where we know that a gene and a compound are related to each other. So for example, when a particular compound targets a particular gene product as an inhibitor, we want to see which of these methods makes that most clear within the data. So most of these projects are just getting underway and in progress, but I'll show you the results of some of our early work here. On the x-axis, I have different staining conditions, and I'm showing on the y-axis 2 different types of scores. One is % strong and the other is % recall. And % strong means how many compounds that were tested in this experiment. We had 90 compounds. How many of them were distinguishable from negative controls? So in other words, they did something to the cell painting phenotype. And secondly, we have % recall. So the 90 compounds are divided into 43 mechanism of action classes. So basically pairs of compounds should look like each other if they're in the same MOA category. And so that's shown in red on this plot. And what we found is that the original Bray protocol was very consistent and is shown here. And our final protocol is a slight bit better than the original, and importantly, it uses fewer reagents. So it'll be cost saving for the partners in the project, but of course also the entire community going forward. So we'll be sharing this protocol. The consortium's mission is to actually share as many of these best practices as early as possible to get really the whole community on board with using consistent protocols, consistent computational analysis, and so on. As another example, we've been optimizing Cas9 lines for the CRISPR experiments, and what's shown here is our test of a number of different A549 lines and U2OS lines all along the x-axis. And for each of them, we've tested at 2 different cell densities, and we're showing again the percent strong and percent recall, but also CRISPR efficiency is added in green. So as you can see, for example, let's start with U2OS. We didn't have very many cell lines here because it was very difficult to get a monoclonal line that worked. In the end, we chose the polyclonal U2OS cells because it clearly had the best CRISPR efficiency, as well as having strong and percent recall. On the A549 situation was much harder to choose because we had a lot of great options here, where we had excellent monoclonal lines that had really strong CRISPR efficiency, as shown across the top here, and we eventually chose the polyclonal line, which gave us really consistent percent strong and percent recall at different cell densities, which means it's going to be a bit more robust. And also what's not shown here is that it very well matched the parental A549 cell line. The parental lines where all the compounds are going to be tested, so having the genetic and the parental cell line, the genetic perturbation line and the chemical perturbation line look like each other in response to perturbations is a really important feature for the overall goals of the consortium. And then lastly, we're aligning public data set with future partner-produced data. And for this step, all the computational analysis is covered. So we're designing computational infrastructure, we're optimizing the pipeline, we're optimizing normalization methods. There will be a huge analysis phase for the last year of the project, where we're first, of course, validating to see that known genes and known compounds that are known to interact or relate to each other actually do match up in the data set. And of course, we plan to discover new findings within the data, whether it's gene-gene connections, compound-compound connections, or gene-compound connections. We also plan to host some public data challenges in order to make this data more structured and nicely organized so that researchers around the world can develop better methods for normalization, for profile matching, and so on. If you'd like more information about the consortium, please sign up for the newsletter. I promise it's not that frequent that we'll send out results, but you'll be first to know about some of the resources we're creating and some of the best practices. The project site is coming within a week or two, so please take note of this web address here, where we'll be adding information in the next week or two to become available. And that includes things like our compound plates, the control landmark sorts of plates that we've been designing and creating plate layouts for. One of them for mechanism of action, which I mentioned, the 90 drugs and 43 classes. And the other is for this target matching exercise, where we have compounds that are known to target particular genes. And in fact, it's pairs of compounds targeting these genes. And the goal is to figure out which methods allow us to match those the best. So those are shown here and will be available, as will be the optimized staining concentrations and protocols that I mentioned earlier. With that, I want to thank you very much. The funding for this project was provided by my Mass Life Sciences Center. Just a tiny fraction of it, the majority of it, is actually coming from all the partners who are listed here on the right. And I have to say, it's an incredible honor to be part of this group of more than 90 scientists that are gathering routinely. We usually have at least 30 or 40 present at each meeting, where we talk about the latest results and get a lot of insight and feedback from experts in the field. So it's really been a thrill to do this project with so many knowledgeable and helpful colleagues across the world. In particular, at the Broad, the colleagues that have been incredibly helpful for this project. First is Naran Chandrasekaran, who's the postdoc on the project, who's been leading a fair bit of this, as well as other members of my lab shown here, and a couple of groups at the Broad who have been doing all the wet lab experiments that have made part of this project. So if you missed any of the URLs or anything you want to take a look at, you can take note of this place where you can get a copy of these slides. And with that, thank you very much for your attention, and excited to see what this consortium produces in the future.